Welcome, class, to our next section here. Today we're going to be talking about glycolysis. Now, I know many of you have already had glycolysis in at least one, and in some cases, several classes. But today we're going to be looking at on this from a little bit of a different angle. And for today, we're going to be setting the stage for being able to predict the chemistry of glycolysis. So let's go ahead and review glycolysis. One, as I wanted to remind you, here's the sort of larger picture of things we're going to be covering in the next couple of weeks of pathways, and glycolysis is this section right here that it fits into. So we'll be talking about that in this interconnection with the other pathways. So as you guys probably remember from your BChem 393 courses, glycolysis has two primary phases. It has a preparatory phase here and a payoff phase. And those two phases here are what allow us to transition glucose from our food sources and then turn them into an energy in the form of ATP. Now, glycolysis does this through the magic of the substrate level phosphorylation that we'll talk about. And as you have probably remembered from your last structure, this whole workflow takes a glucose molecule, it takes two ATPs that come in, and ultimately you get two pyruvates coming out and 4 ATP out. So, and plus 2 NADHs. That's sort of the net set of reactions. So for this course, you should be able to identify every one of the structures, every one of the names, every one of the cofactors for everything you see in this slide here. So if you can pick out the name, the structure, and the cofactor from every one of these reactions, along with the enzyme name, you will be in very good shape. Now, something that I wanted to cover that's sort of it's back along the lines of what we discussed in our last course is that there's some chemical logic here to glycolysis that you may have not talked about before, which is that ultimately we're trying to take this big 6-carbon unit, 1, 2, 3, 4, 5, 6 carbons of glucose, and we're trying to turn it into phosphoenol pyruvate, right? And that phosphoenol pyruvate has 3 carbons in it. So you can imagine that there are two general approaches that you could use for that. One approach would be that you could break this molecule up, and which bond will we break it along? Well, it's always the one that's beta decarbonyl. So if our carbonyl is here, we have alpha, and then beta decarbonyl is right here. So if we were to break it here, you would end up with a 4-carbon molecule and a 2-carbon molecule. So an alternative strategy you might consider is, well, what if we were just being able to move that carbonyl from the carbon 1 to carbon 2? In that case, then the bond beta decarbonyl is right here in the middle of the molecule, and you'll end up with 2 3-carbon molecules. Now, what's the logic behind this? As we all know, glycolysis follows this approach too, right here. So why does it do that? Well, in doing that, it means that all of the steps of glycolysis downstream of that cleavage can be the same. Now, if glycolysis followed this approach 1 that we had laid out, it would have to have one set of reactions for a 4-carbon molecule and another set of reactions for the 2-carbon molecule. So in doing this, life has found a most efficient way to process the carbon using a single set of enzymes. So it's very clever, and it's all and so a lot of the early preparatory phase in glycolysis, as you're going to see, is about moving this carbon from the carbon 1 to carbon 2 to ensure that the bond that we break is beta decarbonyl. Okay, so more on the chemical logic here. So this is I guess more along these lines. Ultimately, we're trying to make this molecule right here, this phosphoenol pyruvate. That's that 3-carbon molecule. So by breaking the bond beta decarbonyl, we'll get 2 of that same phosphoenol pyruvate. Okay, how do we get there? This material should be a review from what you've seen before, but let's talk about it again briefly. So how many ATPs are necessary to make these reactions? Well, you should all know that you have an ATP here in this step, which is hexokinase, and you have another ATP that goes into this step, which is phosphofructokinase, ultimately making this FBP, fructose 1,6-bisphosphate. So, the end product of this preparatory phase is right here, this glyceraldehyde 3-phosphate molecule. Okay, so those are all things that you should be able to identify along with the names of each of these structures. Now, this is proceeds through a number of steps. You know, first step is hexokinase, where the ATP is used to add a phosphate onto carbon 6 of glucose, making this molecule glucose 6-phosphate. There's another version of this molecule called glucokinase, which is found in some cells, and it performs the same reaction. This is a really important enzymatic step because it commits carbon to downstream metabolism, and it's a very highly regulated step in metabolism. Okay, then there's this next step here. This is the phosphoglucose isomerase, and that converts this furanose sugar here, the 6-carbon ring, to a furanose sugar here, this 5-carbon ring. Again, this is getting us ready here for our next phase, where we're going to cleave this thing off into 2 symmetrical molecules. So we're this setup phase where we're moving we're moving these bonds around and getting them ready for that phase. So what's the logic behind this reaction? We're getting ready to set ourselves up for breaking a bond right there. Okay. So phosphofructokinase, PFK1. So is a enzyme that is probably one of the most regulated enzymes in glycolysis. We're going to learn a lot more about its metabolic regulation later lecture. But what it does is it adds the phosphate here back to carbon 1, and it makes this beautifully, nicely symmetric molecule that's already ready for the cleavage there. So on the next stage, this is aldolase, which, as you know, we walked through the reaction mechanism in last class. But this is beautiful because now that we have a fructose molecule, the carbonyl is now on carbon 2, which means that the bond beta to carbonyl, alpha beta, is right in the middle of the molecule. And so the aldolase reaction then results in the production of this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And how can we cleave this carbon-carbon bond? The answer is because it's beta to carbonyl. So what you guys should be able to do right now is you should be able to tell me which of these protons gets abstracted in the first step of this reaction. Now, if you said this proton right here, resulting in electrons resonating here and forming a double bond, and these guys resonating to there, then you'll remember our reaction mechanisms from the last class. So then you have a problem. After you've made this cleavage in aldolase, you have 2 non-symmetric molecules. And as you remember from the logic before, we wanted to have just one set of enzymes. And so glycolysis has an isomerization reaction that converts this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, and it's done by this triose phosphate isomerase reaction. And in doing so, you end up with 2 of the same end product molecules, this glyceraldehyde 3-phosphate, and thus all of the payoff phases of glycolysis can use the one set of enzymes. The other impact that this is going to have that we'll see is that the fluxes downstream of triose phosphate isomerases are going to be twice what the fluxes were upstream of it. And that's because we have 2 input molecules coming from here. We'll learn a lot more about that in our flux balance analysis section of this course. Okay, now we're into the payoff phase. We've put 2 ATP into making this these molecules, and we've now got our glyceraldehyde 3-phosphate, and now we're ready to progress through each of the next steps of the payoff phase. Well, the payoff phase here, as you'll see, produces 2 ATP and 2 ATP here and 2 ATP here, and produces 2 NADHs. So the net production out of glycolysis will be 2 NADHs and 2 ATPs, because we lost 2 ATP that went into forming the original molecule. So what happens to NADH? This is a question. And we'll have to keep this in mind because what happens to this NADH is really important to how metabolism progresses. So we'll talk about each of these steps next. So glyceraldehyde 3-phosphate dehydrogenase, GAPDH, is a really cool enzyme, another well-regulated enzyme. And this is the enzyme that makes glycolysis work. we have a step here that's called substrate level phosphorylation, where phosphate is added, inorganic phosphate is added to this molecule right here, forming 1,3-bisphosphoglycerate, this guy right here. And in the process, we make an NADH. So this is the step, this addition of a phosphate from inorganic phosphate, and it makes this high energy organophosphate molecule. That's what allows us to have these ATP synthetic reactions later on. So from there, we have our first payoff phase here. We're a phosphoglycerate kinase, where we take this 1,3-bisphosphoglycerate, convert it into 3-phosphoglycerate here, removing the phosphate off of this carbon 1 and forming this ATP. So this was this phosphate that was added from substrate level phosphorylation is now being added there to the phosphate, and thus now the energy balance for glycolysis is even on the ATP front, because we had 2 ATP that came in, and this reaction produces 2 ATP when you account for both of the 3-phosphoglycerates that, or the 2 3-carbon units that entered this phase of the reaction series. Then the next stage is that in order to get ready for our final step in converting into phosphoenol pyruvate, we actually have to move the phosphate up to carbon 2. And that'll make sense in the next step. So in order to do that, we have a mutase reaction. As a side note, students should be able to tell the difference between an isomerase or a mutase reaction here. And so what the difference is this phosphate actually goes off into the enzyme, is held in the enzyme, and another phosphate is added on here. So it has the net effect of an isomerization, but it's called a mutase because the other phosphate actually comes off and then is replaced and put on somewhere else. So it's a 3-phosphoglycerate mutase is the reaction here, and that ultimately makes this 2-phosphoglycerate. So the enolase reaction then takes this 2-phosphoglycerate that we just formed, and it makes our very high energy phosphoenol pyruvate. This is the molecule that allows us to form our final set of ATPs in the next step. This is a very, very high energy molecule, and it very much wants to get rid of its phosphate here. Why is that? Well, because you have this carboxylic acid here on this side, and you have this phosphate, and they're both on the same side of a carbon-carbon double bond. So these guys would really like to leave. And so that's what it does. It takes this high energy organophosphate here, takes the phosphate from there, adds it onto ADP. You should be able to do that that reaction mechanism. We'll be working through that in the next class. And that makes this pyruvate here as the leaving group and ATP. Okay. So what to do with that NADPH that we had talked about earlier that was formed in the glyceraldehyde 3-phosphate dehydrogenase step, GAPDH step? Well, we've finished all of our production of pyruvate, and in the process we've made a net of 2 ATP and a net of 2 NADHs. And then you got to do something. You got to convert that into energy. Well, in order to continue to produce pyruvate, the problem is that you're making NADH. If you don't regenerate that into NAD, glycolysis will stop, because that reaction with GAPDH requires NAD, the oxidized nucleotide. And if you've only got a pool of reduced nucleotide, you can't continue to run glycolysis. So we have a variety of ways that we can recycle that NADH. One is through the production of lactate, lactic acid. The other one is through the production of ethanol. And so both of these things have the same net effect, where we have a way of dumping these reduced electrons in the form of NADH onto our final products of glycolysis, pyruvate, ultimately making either lactate or ethanol through these 2 steps. So we'll just walk through those reactions here quickly. So in the case of anaerobic fermentation to lactate synthesis, this happens in human muscles. So this is what you feel when you're sprinting or when you're lifting weights or you're doing any kind of really intense exercise. You'll feel this, the burn that you feel is from this anaerobic fermentation of glucose and ultimately production of lactic acid. So so this is done again to form the oxidized nucleotide NADH in order to allow the GAPDH reaction to occur. The neat thing about this is this doesn't require any oxygen, so it produces the energy very quickly. So this is our preferred mechanism of action in humans and mammalian cells, a lot of organisms, when the need for producing energy fast is what's most important. So when the bear is chasing you, you need to make energy fast. You run really hard. You're actually running faster and burning energy faster than you can actually get oxygen to your muscles. And so your body uses the most energy efficient or the most rapid way of producing energy, and that's just to dump these electrons from NADH onto pyruvate. And then it happens here in this step. You should be able to do these reactions here, and we'll work through them in the next class. But briefly, this hydride here, a hydrogen atom with 2 electrons called a hydride, is ultimately added to this position here on pyruvate, thus making a lactic acid. So in this case, we are taking this NADH, and we are reducing the pyruvate into a lactic acid. And in the process, we're oxidizing NADH into NAD. This regenerates our NAD to allow glycolysis to occur, and then we make this waste product of lactic acid, which is secreted to the blood. So other organisms can use other strategies. Yeast has a slightly more efficient mechanism for converting pyruvate into and regenerating their NADH. And what they do is they take this pyruvate and they decarboxylate it into acetaldehyde, and that acetaldehyde is ultimately then reduced into ethanol. And of course, this is what makes alcoholic beverages, efficacy. This is the main active ingredient in their, beer and wine, that sort of thing, is ethanol. And this is just a waste product of the yeast that are in the fermentation reactions, and they're producing this because they don't have oxygen as an electron acceptor there. So they're using this and allowing them to progress anaerobically and continue to consume the sugars. Through this lecture, you should be able to identify which molecules are the preparatory phase, which are the payoff phase. You should be able to draw all the structures, identify all of the cofactors, and identify all the reaction names and enzyme names. You should be able to do the same for lactate synthesis and ethanol synthesis. And you should also be able to explain, by looking at this molecule here in glucose, explain how this carbon ends up in lactate, ethanol, and acetyl-CoA, and be able to justify that through structures. So we'll be working on those problems here in the next week, and really looking forward to going through some of those exciting new structures.